Hi, everyone. You're listening to Bug Talk, a podcast created by members of the entomology department at Michigan State University. In today's podcast, we're talking to Jean Tsao. Hi, Jean.
Hi there, Sophia.
Thanks for joining us today. My name is Sophia, as Jean said, and hello, everybody. Thanks for joining us today. So, Jean is a professor in the Department of Fisheries and Wildlife here at MSU. And first, we're going to talk a little bit about Jean's background. And then, Jean is a researcher in ticks, in tick biology. And so, we're going to talk a little bit about this timely subject. All right, so let me turn it over to you, Jean. Can you please introduce yourself a little bit?
Yes, well, and thank you very much. I'm so happy to be part of your podcast. As you said, I'm in the Department of Fisheries and Wildlife, but I'm also in the Department of Large Animal Clinical Sciences, which is in the vet school. And I'm not a veterinarian, but we can talk later, part of the background of how I got here. But I am from New Jersey. I was born in New York, moved to New Jersey when I was quite young, grew up there, and then I went to college at Swarthmore College outside of Philadelphia. Then I did my master's and PhD at the University of Chicago in the Department of Ecology and Evolution. But during that time, all my research was done in Connecticut. And I did that through a lab that was at Yale University and continued my postdoc there. And then I came here directly from there to the Department of Fisheries and Wildlife and Large Animal Clinical Sciences. And that was early 2000, let's say, no, 2003. And I've been here ever since.
Cool. So, can we go back a little bit and can you tell us about how you got interested in biology more broadly or research? Where did that begin?
Yeah, so I grew up in the suburbs in New Jersey, and there was a lot of nature, and my parents liked nature, but I didn't, I wouldn't say I did, much. I didn't do any fishing or hunting, but it was a lot of going to natural parks, to arboreta, and just enjoying actually a lot more about plants, and I enjoyed birds and nature in general, but I didn't really know what I was going to do with my life. And I went to college and I really liked biology. So I originally thought, when you have to just write down what your majors would be, I thought biology and comparative literature. And to be honest, I don't even know, I don't think I knew what comparative literature was, but I love literature. And so, but anyway, but the biology department was so great. And it's a small college, so you get to know a lot of the professors, and they don't have very many specific offerings oftentimes.
Mhm.
So you get to learn a lot about various biologies, and enjoyed the labs, etc. But certainly, then I decided, I needed to try to do research. That's what a biology major, is supposed to do, and to enjoy doing science. So my first summer, actually, because I didn't have much experience, I actually was a professional weeder. So I worked as
Oh, that's fun.
I was a horticultural intern and at the Old Westbury Gardens in Long Island. And I loved it. I loved it. But basically, they rotate you among the gardens, but I basically weeded. And I got to learn how to drive stick shift. I got to learn how to operate a chainsaw, didn't kill myself or anyone. So I really liked it. And I liked being outdoors all the time. But I wasn't, I didn't think about necessarily going on in plants and
Mhm.
horticulture. And then the second summer, I was really lucky. I wanted to do ecology, and because I had had a class and I was very interested in it. And it just so happened that I came out to Michigan State. I actually was a research experience for undergraduate, an REU, out at Kellogg Biological Station, and I worked on Daphnia, and I really enjoyed it. Limnology, it was evolutionary ecology, although I actually didn't even know that that's really what the term was at the
Uh-huh.
time. And but I enjoyed the work, and it was really great to be part of a lab and to talk about and science and help the other grad students with their research. I ended up taking a year off of college, and I stayed there at Kellogg Biological Station.
Oh,
So
wow.
towards the middle of the summer, I started asking people, do you need someone to work with you? Do you need someone to work with you? And my mentor, who's not here anymore, was Alan Tessier. I think he got nervous because he thought maybe I'd drop out of college.
Uh-oh.
But it was just that I didn't know what I wanted to do, and I didn't want to waste time
Mm-hmm.
in college trying that was my, my small, I guess, or just my fear at that time. I didn't want to go back to college and not really know what I wanted to do. So I worked actually as a technician for a plant, for grad student who worked on quantitative genetics of a winter annual. And so I got to play with, I
That was
met,
still out at KBS.
was at KBS, and I just forever was so scared that I was killing her plants that she had germinated in the labs with all these crosses, complicated crosses, and she planted in the field in the summer, sometime, and they were germinating. We were measuring cotyledon size, the height of the plant, but they were all tiny. We had to walk across all these plots, and I was just hoping I wasn't killing her experiment. But after that, yeah, I also, again, I was able to then, I was just at that time, the internet was just starting.
Mm-hmm.
And and so, but you couldn't just find out about research experiences. So the other thing I did was, before I left Swarthmore, like there used to be a bulletin board where people who, the biology department must have received ads from people who were looking for, that had research experience for undergraduate programs or internships, etc. So I had taken copies of that, and then I'd come to Kellogg, but I still had that folder. So I was writing people like, can I work in your lab? I'm interested in this. Can I work in your lab? And I had started to think about parasites. I liked the ecology of parasites. I didn't have an interest to go to medical school. I didn't even really think about veterinary school. But I thought, well, I really like ecology. I'm not sure if I want to work on the ecology of Daphnia or aquatic systems. I'm very small-minded, I know. But
Or plants.
and I don't know, I liked all that, but part of me still, I think I wanted to work on something like bugs, like in terms of like bacteria and such, but I didn't want to be necessarily a microbiologist either. And then I thought there must be ecology of parasites.
So
And
how did you end up with the parasites? Like, was that a class that you remembered and liked, or?
I think it was just a unit in ecology.
Mhm.
How, when you think about, and those were macro parasites, but you think about how worms are, some of these trematodes or helminths, that can be transmitted in complex life cycles, and
Mhm.
how they might affect the intermediate host behavior such that the final host. So like how snails, can then, and they'll excrete eggs and such for various parasites. The fish will eat them, and then the fish will become infected with these worms, and they'll swim funnily in a funny way. And then birds will eat them, and then the birds are the final host. So I think all of that, this close symbiosis, that was just interesting to me.
Yeah,
Uh,
I mean, I totally understand. I think anybody that like learns about parasites can not be not interested. It's so fascinating how they have co-evolved.
Yeah. And so I wrote to lots of people, and I even applied at one point for like a Fulbright, scholarship to work on something, didn't get it, but that was afterwards, I guess. But I didn't get anything in that. But there was this one, I saw this ad, and it was at the Harvard School of Public Health, and they had a summer program. So I just wrote them and said, I know you had a summer program, this is obviously not the summer, but I'm an undergraduate and I'm interested in working, getting some lab experience. Is there someone there who might want someone? And that director must have passed my email around, or letter, at that time. And I remember being in the Kellogg KBS mailroom and getting an envelope from this woman, a cardiologist, who said, if you'd like to work in my lab, this is what, I can offer this amount of money and this amount of time. And it was my only offer. I was like, I'm going. and it was not of infectious disease. I wasn't interested in cardiovascular disease, but it was a great experience, because I have to say it's where I learned how to do basic molecular biology.
Mm-hmm.
I hadn't had that as much in college yet, and no labs. And so I learned how to do PCR. I learned how to sequence, and I just learned a lot more about a molecular lab too. And we use radioactivity and all that. And I knew I didn't want to do that anymore in the future. But it was a great experience. And to live in Boston, I really enjoyed that. So I was out here at Hickory Corners in the country. I loved it. Then I got to be in Boston. And
how long were you there for?
6 months.
Mm-hmm.
And then I went back to Swarthmore and finished my last year. And during that time, I applied to graduate school. And then I ended up at Chicago because they were very open with, the type of research you could do, and which is also very different from my current program that I'm a professor in.
Mm-hmm.
But in that program, you just apply to the program, and then they take you, and they hope that you will match with someone eventually.
Did they have like rotations?
They did for the molecular and genetic students, but not so much for the ecology students. But I think what I ended up doing was some readings, like reading, like a independent reading course, I guess, with one or two professors, and in that way, eventually, I guess, determined or asked then, if I could join a particular lab, if they would be my advisor. And so my advisor actually, he's Tim Wooten, and he actually is a marine / aquatic.
Yeah.
And ornithological, I guess. He's an ecologist. And so I think he took me on, maybe out of pity. No, he took me on complex systems, and he was a wonderful advisor. And, but I would, I ended up going off to Connecticut. I made the acquaintance of a professor at the Yale School of Public Health, who was interested in having students doing, the kind of research I ended up wanting to do, field research about the ecology of ticks. But where he was, and he had just gotten set up there, I think most of the students previously who would go to the Yale School of Public Health were much more interested in molecular biology and maybe vaccine development, diagnostics. So I think it was a good time that, I was interested in more ecological research, and he happened to be able to provide a lab to do that.
Mm-hmm, mm-hmm.
And so that it built on that. And I had, worked with Tim to try to develop, let's say, maybe I could work on a system with mergansers and fish and their worm type parasites. And I also thought about frogs and, parasites that they might have that could affect other organisms.
Mm-hmm.
And I thought about hantavirus, but in talking to some other folks, and I thought about Lyme disease, and actually in my undergraduate, I actually did a little, a course, a seminar project on Lyme disease and the ecology of. So I had some familiarity with that. And, but in the end, in talking to the professor at Yale, and perhaps it's his bias, but he said, if you are interested in working on a zoonotic disease, which I had the tendency towards, he's like, you might as well start there. I was really interested in the merganser system, but I thought, okay. And I thought, you can pivot in your postdoc, but that was the advice I was given. And he had said I could work in his lab too. And so I thought about it more, and I thought, okay, I'll do that. And furthermore, with hantavirus, I just thought, do I really want to work in the desert in a spacesuit?
Yeah, and not so much fun.
And a pathogen that might kill you, as opposed to the Lyme disease bacterium, which you can get sick with, but you can take antibiotics, and especially if I know that that's what I'm probably at higher risk for. And so that determined my path, basically.
So did you leave Chicago and go to Connecticut, or you were still based out of Chicago?
So for my academics, I was at Chicago, but then my field seasons and everything, I go to Connecticut. So, in the group I was with, most people, they're field biologists. So they all scattered to wherever their field work was, and I had friends who would go to Papua New Guinea.
Of course.
and wherever, or Tim and his students, the West Coast, and some would come to KBS, right? But I would go to suburban Connecticut.
Wow, exotic.
Yeah. And I had, guess what? At that time, we have oftentimes have, try to have students working with us, and sometimes they're volunteers, or we pay for, basics. I always had a hard time recruiting people. Whereas my friends in Papua New Guinea or Africa or other places, they had no problems.
Yeah, they had volunteering people. Yeah.
Yeah.
So then you started working with Lyme disease, or?
Yes.
Yeah.
For my PhD, it took me a while to figure it out because I did go to Connecticut, learned about the system there, but we were, very much encouraged to come up with our own research project. So I had just read about the whole biodiversity and dilution hypothesis that was first talked about with Lyme disease, where lizards could be potentially diluting the infection prevalence of the Lyme disease bacterium in ticks. And so I thought, oh, that sounds really cool. That sounds really cool, really cool. So I thought, okay, I will go and work where there are lizards. So I went down to the South, and I, through other professors at Chicago, they had told me Savannah River site, which is part of the University of Georgia's system, the Savannah River Ecological Labs. I went there. And I spent a whole summer learning a lot, but it didn't end in fruition with regard to coming up with my research project. And so that was my second summer of my graduate program. Really getting nervous.
Yeah, that's getting tense.
Because in my program, most people, you should be developing your proposal and defending it then, but I had nothing. And then I hightailed it back to Chicago, and I can't remember the timing now, but I think it wasn't until my third year, so, I went back to Connecticut and I really started working. And I think the professor's like, oh, you're back. Aha. Yeah. So then I really developed my research there, which was again still on the idea about potentially testing the hypothesis that a reservoir incompetent host like the lizard could reduce the dynamics of the Lyme disease pathogen in nature. But instead of using lizards, I had the opportunity to use the vaccine for Lyme disease that had been developed at Yale. And
what happened with that vaccine?
Well, so the vaccine is being used commonly now for dogs. It is based on it's a recombinant vaccine based on this outer surface protein, OSA. It was developed in mice, which is how I used it, but it's being given to dogs now. But it had been out on the market for humans, people in around 1998, 99, 2000, sometime around then. But because some people felt that they were getting side effects from the vaccine that were similar to that caused by Lyme disease, I think then at that point, there's so much potential risk for the vaccine producing the pharmaceutical companies that they just themselves withdrew the vaccine.
Mm-hmm.
As vaccine development, they pretty much are in a hole.
That's what I've heard. It wasn't profitable enough, so
and so
we have no more vaccine.
Right, but there is a vaccine that's being now. And so many companies stopped making, developing. Maybe in the back burners they were still working on vaccines, but that they decided not to go that way anymore. But quietly, some pharmaceutical companies were working on it. And so now there is one that's in trials. And so hopefully that in clinical trials. And so hopefully that will come out. That it'll be, effective and such, at least at the population level, and we'll have it hopefully.
Yeah, I actually received, I think, the first 2 doses of this 1990s vaccine.
Me too.
I think I got to the second, and I think I should have received the third, which I never did. So I don't know what status I am right now, but.
So were you were a participant in the trial, or you?
No, this was back in Hungary. We had the vaccine out on the market, like anybody that wanted it could get it. And I am like super attractive for ticks. Like I'm a tick magnet. So I thought, okay, let me do this because I know I'm gonna get it. But yeah, so something happened. I think I traveled, so I never got to the third. But I hope that this one that you're talking about is just like a one off. You can get it.
Well, I don't know actually if it is. And the problem is, actually the original vaccine, they actually thought you would have to continue to have boosters anyway, because the way the vaccine works, it's not something that you would naturally see in nature, because
Oh,
bacterium is so, it's so like many bacteria, it's so interesting. It exhibits, or expresses, I should say, certain proteins on its outer surface when it's in one environment versus the other. So when it's in the tick, it shows OSPA, but when it goes into the host, it downregulates that outer surface protein molecule. It's not needed, and it upregulates these others. But what happens is, because it takes, and this is what's really interesting, it takes 24 to 48 hours for the tick to be feeding, for the bacterium to be transmitted to the human or the host. So during that time, when the tick's sucking down the blood of the host, if the host is vaccinated, it will suck in the antibodies against that protein, and then it'll destroy that protein, that bacterium in the
in the
tick,
tick.
and the tick will not transmit it to the host. So
Ever or just
it
for
won't,
that cycle,
that
feeding
cycle,
cycle.
that's for
Yeah.
that feeding. And so
Uh-huh.
that's why the host naturally will not be exposed to that bacterium and will not
See,
have a natural
that's
booster.
amazing. I didn't know that.
Yeah,
Okay.
well, they just discovered it. I think they didn't know.
So
And they
so I don't have immunity. It will be like the tick and the bacterium that are gonna be
Well,
doing
you would have the antibodies, but what happens is that your antibodies will then go into that tick
into the
and kill
tick.
it along with your complement. But that's what happens. So if you're not exposed to it after a while, that your
The
antibodies.
immune system forgets.
Yeah, exactly. And so that's why they need a booster. And so in a way, it's not the most efficient vaccine. And they thought people would have to get
Yeah.
boosted each year. That's why dogs have to be boosted. And
Uh-huh.
but it is called the transmission blocking vaccine then. And so it's just amazing how it works. But so if you actually so when I was in the field, I had to vaccinate mice 3 times before you could say that they would actually be prevented from becoming infected. Because that's what my experiment was on. I was vaccinating mice, and on the ecological side, I was simulating that they would be like lizards.
Mhm.
But from a kind of a vector control, pathogen control side, the experiment was to see if I could disrupt the transmission cycle and use vaccination of wildlife as a way to reduce Lyme disease risk. So
What was the answer?
well, I could, but the thing was, as a grad student, you're just happy that there was a significant response, right? A
Uh-huh.
significant decline, but biologically, the magnitude of response wasn't great. In
Mm-hmm.
those endemic areas where I worked in Connecticut, about a third of the nymphal ticks are infected.
Yikes. That's
So
a
I
lot.
reduced it to a quarter, about, or less than a quarter. So but if people normally would get at least 3 ticks on them, they had a good chance of becoming infected.
One
I
of them will be.
I reduced it to about 4 ticks. I mean, you have to have 4 ticks. And so if
Yeah.
you're in a place where you get lots of ticks, that's probably not biologically meaningful or
Yeah.
epidemiologically meaningful.
And it would also mean that you have to like vaccinate all these wildlife
Yeah.
that
And these
also
mice.
interact. Yeah.
Right. And then so other people have developed it. Other companies have come in to try to commercialize this. And I have colleagues working on this. And I did it it was a proof of concept way. I actually hand injected, and then you'd hope to catch the same mice coming back to your clinic, 2 more times. But in the lab, we saw 2 shots were just as good as 3. But I hoped that I could get a mouse before it got eaten by a snake or something like that. But the good thing though is the way this vaccine works. Because it is transmission blocking, even if the mouse was already infected, every mouse that I got, even if it were infected, and if I vaccinated it, it would not transmit then to
Mm.
the tick, because the tick would
Interesting.
get the vaccine as well. I mean, the
Mhm.
antibodies as well as the pathogen. So in that sense, for a wildlife vaccine, it was really good, because there's no way in heck that I would be able to beat the ticks out in nature to protecting a mouse hose,
Yeah, yeah.
you
It's
know?
like as soon as it's born, it's probably gonna have
Yeah, as
Lyme
soon as it gets
disease.
out
Yeah.
there, the ticks will be there.
Yeah.
Exactly. And so in that way, this kind of vaccine has an advantage. And so, right. So that's what I worked on
So
then.
are you still working on Lyme disease?
I am, and again, more so on the ecological side. So
I
trying
see.
to understand how it's spreading, but mainly in Michigan, it's the tick itself is what's spreading, and it brings along then the Lyme disease pathogen. So
Can you talk a little bit about that? Like, what's the status?
yeah,
How is the population growing? Like, what are the changes?
sure, sure. When I got here in 2003, Ned Walker, whom
He's in
people
our
audience
department. Yes.
would know, Ned Walker, along with Rich Merritt and others before me that I don't know, but and maybe Howard Russell is somewhat involved, but they had detected the first populations of the black-legged tick, or otherwise also known as the deer tick, in the Upper Peninsula in Menominee County. But it was not known. And then later through Ned's work, it was pretty much determined that in the Southern Peninsula, or Lower Peninsula, I should say, even though sometimes citizens may pick up a black-legged tick or not, it did not seem that they were established down here.
Mhm.
The Lower Peninsula, did not seem to have the black-legged tick. But then Ned, and with a master's student at this, in the early 2000s, who was Eric Foster, who some people might know.
Yeah, I've met him.
So Eric's master's was really cool in that what he and Ned did was that they took the landscape epidemiology map that researchers, including Ned, had developed for the black-legged tick based on presence absence data in Wisconsin, which was ahead of us and is probably the ultimate source of our ticks. They had developed landscape, spatial, ecological map, epidemiology, depend on which side of if you're entomologist versus epidemiologist person. What they found was certain habitats were more associated with the ticks than other habitats. And they took that model and placed it on top of Michigan. And so then they went to look for the tick. And also they were strategic. I think they had known that there were some ticks in Northwest Indiana already. And so through Eric's work, he found established populations using the models and just general, a good guess of where the source could be. They found established populations in Southwest Michigan. And
Yikes.
yeah, so I came in 2003. Eric was still a student. He was writing that up. And then Sarah Hamer, or Sarah Yermich, when she first came, she was a grad student. And along with Graham Hickling and Ed Walker, then we built on Eric's work. And we wanted to be able to monitor where this tick was spreading. And so we set up 2 large transects, one along the coast, because coastal habitats, sandy soils, upland forests were indicated as really suitable habitat, according to Ned's and others' models. And then had another transect coming up into Lansing, diagonally from Southwest Michigan. And so Sarah's PhD, she hit the ground running. She's an excellent grad student, but even so, 5 years, and it's a 2-year life cycle. And I was thinking, I don't know if this is the best project for a grad student. But Sarah decided to go on to get her DVM as well. So she was here about 8 years. And so during that time, we were able to see how the black-legged tick spread up along the shores of Lake Michigan, along those coastal forests. And they kind of were coming inland in the eastern fashion, but just much slower. But, in the beginning, when we showed the data, makes sense, because research hadn't been conducted in an active, rigorous manner previously to document their presence going up the shore. I'm from New Jersey, we call it the shore, but Lake Michigan, you don't know just because you find the ticks whether they were there already or if they're moving, right?
Mhm.
So, but with 6 years of surveillance, it really did seem that we were seeing increases of tick abundances in areas where we had found them, but they were increasing, and we were seeing them move north.
Spread.
Yeah, and then really I've continued that work with other grad students, and we've certainly really confirmed that pattern. And then they started moving inland. And now, so that was 2004.
Yeah, let's look at 2021 now.
Yeah, I was going to say, so Sarah began 2003, 2004, picking up on Eric's work. By 2010, they had gone up to just reach Sleeping Bear Dunes. All right.
Mhm.
For, people up here, and they had kind of come in here, kind of like maybe just the beginnings at Lux Arbor, which is Kellogg Biological Station. But then in 2010, for Custer, which was one of our sites, we started seeing a tiny bit more. By 2014, it was a lot more. And Eric now had moved over to the health department. And so he got lots of information from the public, and also Howard Russell gets information from the public. So we have this, network of people telling us where ticks might be. And for example, by 2014, Fenner Nature Center started to be colonized, or they were already colonized, but we've seen their numbers, that's right here in Lansing, just go through the roofs to 2021. And similarly, in the beginning, we saw a little bit a low infection prevalence in the ticks for the Lyme disease bacterium, but then we couldn't find it for several years, but now they're kind of creeping up again.
Uh-oh.
Yeah.
So, I remember a few years ago, I had these tick collecting kits in my lab. Are those still around? And is anybody collecting those? Like, I haven't seen them in circulation forever.
Right. So the health department, MDHHS, they would provide these tick collecting kits as part of what we call passive surveillance. So because the health department does not have funding to have all across the state for there to be active surveillance, so looking for ticks, in a systematic way, a great way would be through if citizens found ticks and submitted them. And then the black-legged ticks would then get tested for the pathogen so that they would know whether or not pathogens were in the area as well. And then therefore the health department can use that information then to inform the local health departments and the city and the public. But now you can still order these tick submission kits from the health department. They can still send them to you, but they no longer test for the pathogen.
So what do they do?
So they will just identify that the tick, what species.
It's like black-legged tick.
Yep, exactly. Or American dog tick, or the Lone Star tick, which is just invading Michigan as well.
Is that just a financial state that they're in that they can't test them?
Well, that's partially that. It happened, the first time they decided not to test was last year with COVID, because the labs were just inundated. They had to be testing for COVID. There was no more bandwidth for other tests. But I can also tell you that from the side of the CDC and also the Infectious Disease Society of America, which writes the guidelines for the prevention, diagnosis, and treatment of Lyme disease, it's not recommended that if you find a tick on yourself to have it tested. And it might sound counterintuitive, but this is the rationale. First of all, part of it is when they did epidemiological studies in the US as well as in Europe, the people who actually find the ticks on them, even though the local infection prevalence of the ticks might be 10, 14, 20%, and therefore it should be the same probability, basically, when you think about a tick that's biting you, most people who find the tick, and I wouldn't say most, but at least half the people who find the ticks, they find them before that 36, 48 hour time period. So the ticks will not have transmitted the pathogen. All right. And then if people decide to treat them, then there are side effects to being treated with doxycycline. That's one of the main antibiotics. So health professionals don't recommend just giving out antibiotics, especially in places where there's a high risk of contacting these ticks frequently. All right. Number two, there are some false positives and false negatives, that one always has to take into consideration when a tick is biting you, or that, testing a tick for pathogens. And also so you may be lulled into a false sense of security, or you may have unnecessary concern
Treatment. Yeah.
concern and treatment. And the thing is, sometimes the tick that you find is not necessarily also the tick that could infect you.
Yeah.
So you might find a tick and it's not infected, but you didn't find the one that actually infected you. And most people never see the tick that feeds on them, even though they will feed for 3 to 5 days. But it's
How do you not feel them? I mean, they're pretty big when they're full of blood.
well, but the nymphal stage, which I have some around, but they're small. They're the size of poppy seeds in the beginning, and they may be behind your knee, they may be in your groin, your belly button, your hairline, and they will get larger, like you said, as they feed. But sometimes, especially like if you're older or something, you might not be able to see it, and you might feel it, and you think, oh, it's just a scab from something. And so these ticks, as opposed to mosquitoes, they come in, they feed, and then they pull out for within seconds and they're gone. Ticks have to feed for so long, and so over evolutionary time they have developed all these molecules, proteins in their saliva to really quell the immune system, because they are, a foreign object. And so your immune system immediately will be trying to attack it. It should be itchy. There'll be inflammation and all that. But there isn't, unless you get exposed to ticks frequently. And so I actually will develop an itch response to
Me too.
and that's really good for us.
Oh, okay.
It's our warning system, and besides checking, but normally that doesn't happen, in the beginning when you're first, being fed on by ticks.
Yeah. So I want to be respectful of your time, so we're gonna wrap up here in a few minutes. But can you just summarize the tick situation this year, and what are we looking at in terms of Lyme disease or chances of getting Lyme disease here in Michigan?
Okay, well, first of all, so I left this off at 2014, but we jumped to 2016. And it's really interesting because the black-legged tick, at least with southern Michigan, has spread basically over to Lake Huron. All right. So in the thumb area it's at Lake Huron, but it's patchy with its distribution, though. It's not as, the highest risk is probably still closer to Lake Michigan. And then there are patchy areas where the risk will be higher or lower. But certainly within the last few years, Ypsilanti and Ann Arbor became areas where the ticks have been found and infected. But it's all the way over to Lake Huron. Going up the coast on Lake Huron side, they're also there. And I don't know, a hypothesis is perhaps birds have been dropping them off along Lake Huron. And because the lakes, the weather conditions, climate conditions, I should say, are more hospitable to ticks, that they are able to survive better. Whereas in the middle of Michigan, we have been surveying, and we do not, the northern lower, we have not been able to find as many black-legged ticks. I don't know if it's too dry, not enough snow cover, not enough habitat. There's plenty of deer and small mammals. So eventually, and the UP similarly, the east side is getting colonized, but it just hasn't, not as many black-legged ticks are over on the east side as the west side. But I think overall, this tick is a generalist tick, and this pathogen overall is a generalist pathogen. So once it gets to a place, there's gonna be Lyme disease risk. And there's no reason why Michigan will not be like Wisconsin and the eastern part of Minnesota. We will have it. And other pathogens are following in the wake. Dog ticks this year, they were exploding, and dog ticks are generally found throughout the state. Although Michiganders have told me that where they've lived, they've grown up there weren't ticks there before, now there are. So we don't study the dog tick as much, so I can't really say, what might be going on. But I think just climate change, land use change, changes in wildlife, abundances and distribution, those will affect where ticks will be. And then, Rocky Mountain spotted fever is the main pathogen the dog tick transmits, but it's very low prevalence and very rare here in Michigan. But this year with
Good.
all dog ticks, I would still say to people, I mean, no one wants to tick on them anyway, but everyone should watch out, and maybe physicians should look at, be wary that we could have more Rocky Mountain spotted fever. It's just a prediction. And the Lone Star tick, which is from the southeastern US, a very aggressive tick, also carries many pathogens, but it's usually at low prevalence. They have snuck into southwestern Michigan as well. We can blame Indiana again, but they're here. They're established in Berrien County, very focal, but they are associated with the red meat allergy. And so, I think a lot of people are probably more upset at that than necessarily pathogens. But so those are the main human biting ticks, and people, it's the same precautions. Just stay on the trail, use a repellent, check yourself often, and then always save your tick, because if you do get sick, different ticks transmit different pathogens. So it's very good information to be able to provide your physician what it is. And if you don't know what it is, you can use our mobile health app called the Tick app.
Oh, I'll link to that.
Yeah. And then also, but the state health department, that's another reason to send the tick in. They'll identify it and they'll have that record, et cetera. So, but save the tick, don't flush it. I know we want to, but there's evidence there that can help you.
Yeah, that's great. That's great information. Even if you like yank out the tick and the head stays in there, it's still identifiable, right? It's
for the most part. Yep. Yeah.
Okay.
It's worth keeping. I mean, so you bring a good point though. I mean, bring up a good point. Try to get the tick as close to the mouth parts in the right touch of the body. But I know it's hard sometimes. So
yeah. Well, ticks are good news all around, aren't they?
Yeah.
Job security for you.
That's right. But unfortunately, I always do want people to still enjoy being outdoors, right? And it's just like we wear a bike helmet for bicycling, just wear your repellent and still just enjoy being out there.
Well, thank you so much, Jean, for coming on and sharing this with us. I'm sure it was useful, and I'll make sure to link in the show notes to some of these resources that you mentioned and shared with us. And I think we should have you back more often so that you can let us know about the latest and greatest we should know about ticks.
Anytime. Yeah. Thank you so much for having me. And I hope that this podcast goes on for many, many more seasons.
Well, we're trying our best. And thanks everyone for listening, and we'll see you in the next podcast.